Okay, so we're going to go through the structures associated with and the different cell forms and embryo forms associated with the pregnancy and development lab. And so just to give us some orientation, we have the uterus here. Okay, here's the cervix, the vagina. Here's the uterine tube, the fimbria. Here's an ovary with all the different stages of the ovarian cycle within it. And then we have an ovarian ligament right here that's connecting the ovary to the uterus. Okay, so we have an oocyte that's being ejected here during ovulation, okay, from a vesicular follicle. And the oocyte will be swept into the uterine tube by the fimbriae, because they'll be sweeping over the surface of the ovary. And the oocyte, which is in metaphase of meiosis 2, is going to start meandering slowly down through the uterine tube. Okay, now from the time that the oocyte is ovulated, it only has 12 to 24 hours of viability. So if it doesn't meet up with a group of sperm in that amount of time, fertilization will not occur. The oocyte will degenerate, and that's pretty much it for the oocyte. Okay, so fortunately for us, our oocyte is encountering a sperm here, okay, and there's a larger representation of that up here in B. Okay, now this is really an urban myth, because we can't just encounter one sperm, we have to encounter thousands, because there is a thick shell of glycoproteins and smaller cells around the oocyte, so we need thousands of sperm to drop their acrosomal contents, the special lysosome that they have at their front tip, onto the surface of that oocyte to break through all of that covering. Okay, so after that 1,000 or so sperm push up against the oocyte and have their acrosomal contents deposited, then eventually you'll get through enough of it that one of the sperm will be able to fuse with the membrane of the oocyte. Okay, and once that happens, then fertilization can occur. Okay, so the cell that's formed from fertilization is the zygote. There's not really a representation of that on this image, but you have one in your lab manual and in your textbook. Okay, and the zygote is the original cell that's formed at fertilization. And after the zygote is formed, it will start going through divisions, and those early divisions, each one is called a cleavage. Okay, so the zygote will divide into the 2-cell stage by the first cleavage, then into the 4-cell stage. And the 4-cell stage is pictured here in C. Okay, and that would be from the second cleavage, and then we'll just continue having cleavages and increase in cell number. Once we get at least 16 cells, which is about 36 hours into the process, then we have this stage, which is the morula stage. Okay, now the morula stage is still going to be in the uterine tube, still making its way to the uterus, and it will be a mass of cells. You can't tell any head end, any tail end. All the cells look roughly the same size, no distinctive features on them. And it's called morula because morula in Latin means little berry. Okay, so if you think about how a blackberry or a raspberry looks, that's about how this stage is going to look. Okay, so we're going to continue dividing, but now inside the embryo, we're going to start seeing spaces form. Okay, and we're going to start seeing obvious signs that some cells are going to have different fates than others. Okay, so when we get actual spaces inside the embryo like you see here, then we get to the blastocyst stage. Okay, so in the blastocyst stage, we have a place where the embryo is going to develop, and then we have these spaces. Okay, one is where the amniotic fluid is going to accumulate, and this would be the amnion here. And then we also have this area here, which is the edge of the yolk sac, and the yolk sac material will be in here. Okay, and the blastocyst stage is the stage you have at about 3 or 4 days, and this is the stage at which the embryo has reached the uterus and has moved into the lumen of the uterus and is now looking to attach to the endometrium. Okay, and it's this stage where we start to see the formation of the trophoblast and the specialization of the trophoblast into the syncytiotrophoblast and the cytotrophoblast, where the syncytiotrophoblast is going to start degrading the uterine wall and allowing the embryo to burrow into the endometrium. Okay, so by the time that process is finished, the endometrium should basically close over the embryo so it's right within the wall of the uterus. Okay, as development progresses, we go through a gastrula stage. That's not pictured here, but that was the one where you had the 3 different germ layers present for the first time. So you may remember that from your textbook. And then we're going to start developing more and more, and the embryo is going to become more apparent. Okay, so this, now we've jumped pretty far here to a 21-day embryo. Okay, so we have the head end up here, the tail end down here. That big red thing is the heart. Okay, there's a start of what's going to be the location of the nervous system here. Okay, and on this embryo, we can actually see all 4 of the extra embryonic membranes. Okay, so we have this one here, which is the yolk sac. Okay, and all the yolk would be in here. You can see that this is actually attached to the anterior abdominal area of the embryo. Okay, so when you talk about this having stem cells that are going to come into the body and how it's going to form part of the digestive tract, that makes sense when you see where the yolk sac is attached. Okay, so the stem cells that you have in here will go into the gonads eventually to allow us to have stem cells for sex cell production. They'll also go into the bone marrow eventually to give us the ability to produce our different types of blood cells. Okay, we have the amnion here, and the purpose of the amnion is to produce the amniotic fluid, which is going to accumulate in this space. Okay, and the amniotic fluid has several functions. It helps to regulate the temperature around the embryo. It acts as a place where the embryo can dump some excreted materials, and it's cleaned up by capillaries in the amnion. It's also important because it keeps even pressure on all sides of the embryo. Okay, if the embryo was in a space where it was getting pushed, you know, it's pretty soft at this point. It would grow in a really misshapen manner. Okay, so the amniotic fluid makes sure that you have nice even pressure all the way around. Okay, and extremely protective to the embryo as well. Okay, the little thing that you see here, indicated by the 35 and the arrow, is the allantois. The allantois is the extra embryonic membrane that will form part of the urinary bladder. That makes sense. We're at the tail end of the organism here. and it also forms part of the umbilical cord, which is starting to develop right here. Okay, and then last but not least, we have the chorion. The chorion is what develops from the cytotrophoblast. Okay, and then you can see the chorionic villi out here, and the chorion and the chorionic villi and the capillary beds within there are what become the fetal portion of the placenta. Okay, now here we have a 5-week embryo. We're a lot more developed at this point. We have a distinct head region. You can see where the different swellings for the parts of the brain are going to be. There's an eye. Okay, we've got some limbs that are apparent. We still have a little bit of a tail, but don't worry, we'll get rid of that. See a nice umbilical cord. Unfortunately, they made a mistake with the illustration here. This large umbilical vein should be red. The 2 smaller umbilical arteries should be blue, because the umbilical vein is carrying the oxygen-rich blood to the embryo and fetus. Oxygen-poor blood coming back to the placenta through the umbilical arteries. Okay, now this is the next panel we're going to move in. We're going to go through embryonic development and finish that up. So this is actual size for a 6-week embryo. Here we have an 8-week embryo, and this is the end of the embryonic period. Now we're moving into the fetal period. So we have 10 weeks, 12 weeks, 14 weeks, and then in this larger illustration, the 16-week fetus. Okay, so externally at least, it looks like our fetus looks pretty much like a baby. Okay, internally there's still some organ formation and fine-tuning that needs to occur, but outside it looks pretty good. You know, this looks just like a very small human that needs to grow up so it can survive on the outside. And to a large extent, that's what needs to happen. Okay, we can see the uterus here very well. Okay, the endometrium isn't quite as obvious, but you can definitely see the myometrium very easily with all that smooth muscle. Okay, and then the perimetrium would be our layer closest to the peritoneal cavity. Okay, cervix, still nice and thick. Okay, my thumb is right now in the urinary bladder. I'll move that out of the way. Okay, the amnion is all around the fetus. Here's the umbilical cord, and here's the placenta, and this would have both the fetal and the maternal portions within it. We can't really distinguish them at this point. We're already noticing that the fetus is starting to take up a good amount of room, so organs are being moved out of the way to accommodate. We'll see that even more in the next section.